Epigenetic reprogramming is a revolutionary process in molecular biology that aims to reset the epigenetic landscape of a cell, essentially wiping or modifying the chemical modifications that regulate gene expression without altering the DNA sequence itself. These modifications, including DNA methylation and histone acetylation, act like molecular switches that determine which genes are active and which are silenced in each cell type. Over time, due to environmental exposures, stress, inflammation, or aging, these epigenetic marks can become dysregulated, leading to cellular dysfunction, aging, and disease. By reprogramming the epigenome, often through the controlled expression of specific transcription factors known as Yamanaka factors, OCTOBR4, SOX2, KLF4, and CMYC, scientists have been able to revert differentiated adult cells back to a pluripotent, embryonic-like state. More recently, researchers have shown that partial reprogramming, where these factors are applied for a limited time, can rejuvenate aged cells and restore youthful function without erasing cellular identity, a promising step in reversing aging and tissue degeneration. In animal models, partial epigenetic reprogramming has led to restored vision, improved muscle regeneration, and increased lifespan. This powerful technique is now being explored not only for anti-aging therapies, but also for treating neurodegenerative disorders, cardiovascular diseases, and even cancer. However, the process is still under intense investigation, as uncontrolled reprogramming carries risks such as tumor formation or loss of cellular identity. As our understanding deepens, epigenetic reprogramming stands at the forefront of regenerative medicine, offering a glimpse into the future where biological age may be reversible.